Hello, everyone, and welcome to today's webinar, Solutions for Dendritic Cell Research. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Our speaker will answer questions via the email address you provided during registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Nizar Yousif, Global Product Manager, Immunology, Immuno-Oncology, Miltenyi Biotec. Dr. Yousif, you may now begin your presentation.
Welcome everybody to our webinar, Dendritic Cell Research Solutions. In this webinar, you will learn about Miltenyi's tools and methods for studying disease subpopulations in human and mouse. Here's the agenda for today's webinar. First, we will have a brief introduction. Then I will talk about sample preparation for dendritic cell analysis. Afterwards, I will talk about our flow cytometric analysis solutions for human and mouse DCs, followed by magnetic isolation of DCs and more DC generation solutions. And at the end, I will tease you with our spatial biology platform for DC characterization. Now, the introduction. Dendritic cells were discovered by Paul Langerhans in 1868, and their function was described by Ralph Steinman in 1973. So these cells are professional antigen-presenting cells that on one hand promote immunity to foreign antigens, and on the other hand are capable of inducing tolerance to self-antigens. Dendritic cells can regulate the immune response by secreting pro and anti-inflammatory, cytokines. Yeah, and these cells come in different flavors and can be differentiated based on different factors, including the expression of cell surface markers, tissue localization, ontogeny, and their functional specialization. This is a simplified overview of dendritic cell subsets in human and mouse, and three major DC subsets can be found: myeloid conventional DC1, CDC1, myeloid conventional DC2, CDC2, and plasmacytoid DC, PDC. And here you see the monocyte-derived disease, which do not share ontogeny with conventional disease and can be derived from monocyte lineage during infection and inflammation. So, and due to time reasons, I will mainly be discussing the DC subsets listed here. However, for learning more about further DC subsets, I encourage you, to have a look at the publication, listed here on this slide, and further recent, related, topic related publications. As mentioned, DCs are specialized antigen presenting cells and have essential roles in initiating and regulating innate and adaptive immune responses. However, these cells are basically different in many ways. Here I will focus on key features of human DCs. So DCs have different phenotypical features. They express various type of markers that can be used to identify these cells. Among the most commonly used markers, CD141 is mentioned for CDC1 and CDC1c, is expressed in CDC2, and markers like CD123, CD303, and CD304 are commonly expressed in PDCs. Yeah, so DC subsets have different ontogeny and depend on different growth factors. Yeah, so their identity is regulated via the expression of different transcription factors. And functional specialization of DC subsets results from different, from the expression of receptors, different receptors, including, the pattern recognition receptors, yeah, such as PRRs, yeah, but also various cytokines secretion. Yeah, so it's commonly known that CDC1 are potent inducers of Th, helper T cell 1 immunity, and CDC2 can induce Th1, Th2, as well as Th17, and PDCs, initiate antiviral responses. So to learn more about human and mouse disease subsets, I recommend you to read the excellent review by Stephanie Vuklek. Yeah, this is, this paper is published in Nature Review in Immunology. Yeah, so I recommend you to read it for further information about human and mouse disease. Next, I will talk about sample preparation for dendritic cell analysis. Sample preparation for DC analysis can be challenging. Why? Because we have many tissue sources and there are different protocols available, which can be overwhelming to select the right one. So researchers working with human DCs most commonly use blood and blood products such as Buffycoat, LRHCs, and leukopacs. Here, sample preparation for analysis is easy, and researchers often perform density gradient centrifugation or RBC lysis. Human bone marrow samples are usually purified by density gradient centrifugation and RBC lysis, whereas in mice, researchers usually use femurs and tibias and flush the bone marrow with medium or buffer. So the analysis of tissue-resident infiltrating disease is most complicated due to tissue-specific enzymatic digestion and dissociation protocols. Our sample preparation solution for Miltenyi Biotec that can, make your life easier here are the GentleMax tissue dissociators, yeah, and the GentleMax tubes and tissue dissociation kits. So to simplify, the labor-intensive process of preparing single-cell suspension for DC analysis. Yeah. So using this technology, viability, epitope preservation, reproducibility, and speed are guaranteed. This is an exemplary analysis of mouse DC subsets after GentleMax dissociation of spleen, lung, and lamina propria. So enzymatic tissue dissociation with the GentleMax is a gentle method to obtain a suspension of single cells for further downstream analysis. Yeah, so the enzymes including at Miltenyi's kit are highly purified to maximize epitope preservation. The enzymatic digestion and tissue-specific mechanical dissociation parameters are optimized to ensure high cell viability, lot-to-lot consistent enzymatic activities and automation and standardization, ensure high reproducibility. So you are well set here. Now let's move to the next part, flow cytometric analysis of human and mouse disease. Flow cytometry is one of the main and essential tools for studying dendritic cells. Yeah? So if you're trying to accurately analyze cells by flow cytometry, the most reliable solution we can offer you is our REAfinity antibodies. Yeah, these are recombinantly engineered antibodies, yeah, in which the FC region has been mutated to minimize background signal. Yeah? So if we translate this to the myeloid cell, language, yeah, and we know that the dendritic cells belong to the myeloid cell family, yeah, the FC blocking step is not required anymore. Yeah, so the REAfinity antibodies show high, epitope specificity, and low background binding. Yeah, and all so and all the REAfinity antibodies, are IgG1 isotype, yeah, IgG1 isotype. So making, yeah, the isotype control staining easy. Yeah. So the REAfinity antibody ensure, you consistent and reproducible results. So you don't need to worry about, about consistency, and reproducibility, of your experiments. In this table, is an optimized panel for analysis of splenic disease using MaxQuant or similar flow cytometric analyzer. So the gating strategy starts with the selection of cells and the exclusion of debris using side and forward scatter. Yeah, and next, you select singlets and live cells where viability dyes such as PI or 7AAD can be used, and whenever is possible, so we recommend the macrophage exclusion, yeah, using, for example, the markers F480 and CD64 staining, yeah, and finally we can analyze the abundance of the three major disease subsets, in the mouse spleen. So the conventional disease show high level of CDC, CD11c and MHC class 2 expression, and can be divided into two, major, subsets. Yeah, this is XCR1 expressing CDC1 and SRIP, or CD11b expressing CDC2. Yeah, so the plasmacytoid disease usually express, intermediate level of CD11c, thus single, cyclic H and PDCA1 staining is recommended, to facilitate more, precise gating. This is similar to the splenic DC characterization approach I described on the previous slide. After dissociation, mouse spleen cells were stained with the antibodies described here in this panel. Yeah, and after exclusion of dead cells, debris and doublets, yeah, so CD11c positive and MHC class 2 positive conventional DCs were selected and further subdivided into XCR1, yeah, and CD172, yeah, or CD11b, positive subsets. And plasmacytoid disease, yeah, plasmacytoid disease can be easily identified by cyclic H, yeah, and PDCA1 expression. So, yeah, you can find detailed staining protocol under the link provided here, yeah, on our website. So Miltenyi Biotec website. When you get the PDF of this PowerPoint, yeah, you can, you can click here, yeah, on the hyperlink, which will bring you directly to the detailed protocol on our website. In the previous slides, I talked about the analysis of mouse DC. Now, to analyze the abundance of the main three disease subsets in human blood, we have developed a staining panel and gating strategy in collaboration with Jolanda de Vries from the University of Nijmegen. Yeah, so after selecting viable and CD45 positive cells and the exclusion of CD20 positive, yeah, this is the B cells and CD14 positive monocytes, yeah, the 3DC subsets can be identified where cross-presenting CDC1 co-express, these markers, yeah, and CDC2 express these two markers, and plasmacytoids, DCs express, these two markers, yeah. And this is actually what you see here, is nicely depicted, here in the flow cytometry, plots. In this slide, you observe a detailed step-by-step getting a strategy in order to evaluate DC abundance in blood samples, yeah. So cells were lysed, cells from lysed blood were characterized using the panels shown here in this table, yeah. First the debris, and then the 7AAD positive dead cells were excluded, and then CD45 positive cells, CD45 positive cells were selected, yeah. So, and from there, yeah, so side-scatter low cells were selected, and yeah, so the CD14, CD20 negative cells were selected, and within these, the abundance of PDC, expressing CD123 and CD303 were selected, yeah, as well as, CD141 expressing CDC1 was evaluated, yeah. So, and next, after excluding, the CD123 positive cells, the abundance of CDC2, were assessed, using these markers. So CDC2 is positive for these two markers. So you can find detailed staining protocol, under the link provided here, on our website, yeah. In the next slides, I will talk about our magnetic isolation solutions for dendritic cells. In order to perform molecular analysis of cells, or to perform cell culture experiments, researchers need to isolate, the cells of interest. And so how do they do that? Yeah. So we have here our solution is the magnetic cell separation. So it's simple cell isolation method. Yeah, it enables the preparation of high quality cells, suitable for downstream applications such as DC and TC, T cell co-culture, what they call, MLR, mixed lymphocyte reaction, or some people call it mixed leukocyte reaction. So using this technology, reduces time required for flow cytometric analysis or sorting rare, cell subsets. So, and as you observe here, yeah, in the flow cytometric, analysis, shown here at the bottom, yeah. So the abundance of resident and migrating DCs in mouse lymph node cell suspension, were more than 60-fold enrichment, of target cells, yeah, using, our CD11c, microbead ultra, ultra pure. So, and the max cell separation technology is compatible with most laboratory techniques and applications. The CD11c MicroBeads ultra pure mentioned on the previous slide were used to isolate CD11c positive cells from spleen, lung, lamina propria, and tumor tissue. Yeah, so tissues were harvested and dissociated using tissue-specific dissociation kits and the GentleMax dissociator. And the cell suspension was labeled with CD11c microbeads ultra pure and magnetically separated. Yeah, so followed by flow cytometric analysis to compare the abundance of dendritic cell subsets in the starting material and in the enriched fraction. Yeah, and in the so the bottom panel beautifully shows 10, to 42-fold enrichment of dendritic cells in the tissues, tested. At Miltenyi Biotec, we have many products and solutions for optimal enrichment of mouse disease. Yeah, for more information, please visit our website or contact your, local team, sales representative to find the products that are tailored to your needs. In this slide, you observe an example of enrichment of human cross-presenting CD141 disease. Yeah, so cell isolated, the cells isolate. This is isolated with CD141 microbead kits are highly enriched. They express typical CDC1 surface markers such as CD141, HLA-DR, and co. Yeah, these cells are fully functional upon stimulation with TLR3 agonists, yeah, such as poly-IC. They're able to express, upregulate expression of activation markers, CD80, CD68, and co. These cells are also able to secrete typical cytokines, yeah, so GM-CSF, yeah, interferon gamma and alpha, TNF alpha, and other various interleukins, as you observe here. We also have many solutions for isolating human disease. Yeah, as mentioned before, you can visit our website or contact your local sales representative, to find more about our DC, human DC products tailored to your needs. Now, let's talk about more DC generation. As the name says it, monocyte-derived disease, so the generation of monocyte-derived disease begins with the isolation of monocytes, yeah? So the solutions presented here are developed to facilitate the isolation of total, so they call it also pan-monocytes, yeah? So CD14 positive classical monocytes, CD14 positive classical monocytes, and CD16 positive, CD14 negative non-classical monocytes. Yeah. However, for more DC generation, researchers use either the CD14 classical monocytes or pan monocytes isolation. Yeah. So the CD14 microbeads are the most commonly mentioned and cited solution, for the generation of more DCs. As highlight product, let me introduce you our straight from solution for monocyte isolation. Yeah? So straight from technology enables the separation of highly pure cells, directly from different, blood sources with all the benefits of the MACSima Imaging, yeah, as well as easy and fast enrichment of leukocytes. So no need for density gradient, no cell counting, no washing, and no erythrocyte lysis needed. So is the efficient solution for monocyte isolation. We can support you with the complete workflow for more DC generation. So one of the key elements, one of the success key elements for that is the standardized culture using max premium grade cytokines. Yeah. So to optimally differentiate and match your MODC, we know the exact unit per milligram dosing, which allows high reproducibility. So this data, yeah, show the functionality of MODC differentiated and matured using, our cell culture solutions. Yeah, these cells are able to increase the expression of activation markers during maturation. They are able to migrate in a, trans-well migration assay, and they are able to, to secrete cytokines, yeah, so such as IL-12 and IL-10 upon stimulation with, ligands such as CD40 ligands. Yeah. So, and you find the detailed protocols, yeah, about MODC generation and additional details for downstream applications, in this link, yeah. So in our website. In the next two slides, I would like to tease you a little bit with our spatial biology platform, which allows you to characterize dendritic cells and gain spatial information about these cells. For spatial analysis is the MACSima Imaging System, which is part of a complete imaging platform, that enables you to analyze hundreds of markers on a single sample, and in a fully automated manner. So it means apart from loading the samples and the reagents, no manual steps are involved after starting, the experiment. So moreover, all the necessary components to perform such experiments will be provided, which are sample carriers, antibodies, and also the analysis software. So I would like to mention here that the MACSima Imaging Platform is an open platform with regards to antibodies, which means that you are not dependent on the antibodies Miltenyi Biotec is providing, although we have a huge portfolio of high quality recombinantly engineered antibodies. So we have the largest antibody portfolio compared to all other multiplexing technologies on the market right now. So this complete platform solution therefore opens up such complex analysis to a very broad spectrum of users as the technical hurdles are really very low. So it's also very flexible as it can handle any kind of fixed samples, such as tissues, adherent cells, or suspension cells. And the preparation of the sample does not require laborious and complicated steps, which means effortless experiment preparation. In this experiment, the tumor immune microenvironment, particularly the tertiary lymphoid extraction of ovarian cancer tissue, was investigated using the MACSima. Here you observe an overlay of 7 markers, characterizing the tumor cells in green and various immune cells, such as B cells in violet, T cells in blue, plasma cells stained with CD138 in white, dendritic cells, stained with CD68, with CD86 in brown, and myeloid cells stained with CD14 in yellow, and endothelial cells, stained with, MACA79. So thanks to the, MACSima platform, for analyzing, these several markers in the tumor immune microenvironment. Our goal is to support your DC research from A to Z. So from sample preparation to cell separation to flow analysis to cell culture, as well as DC characterization using our spatial biology imaging technology. We have available material online, such as protocols and application notes that can support your DC research. Please feel free to visit Miltenyi Biotec website and check that, or contact your local sales for further support. This is my disclaimer. Thank you very much for your attention. Now, if you have any question, please submit it and I would be happy to answer it.
Thank you, Dr. Yousif, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Questions will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.